2014, a California groundskeeper was diagnosed with non-Hodgkin lymphoma. He had spent years spraying a weed killer on school grounds. The label said safe. The company said safe. The molecule attacked a metabolic pathway humans don't possess, the Shikimate pathway. But his body found another way to take damage. His case became the first to force a jury verdict behind closed doors. This is the story of a molecule that was supposed to vanish from the body, yet reappeared in breast milk, in umbilical cord blood, in the urine of children who had never touched a sprayer. A molecule sprayed on lawns, fields, schoolyards, until the evidence accumulated in court dockets and cancer registries. Welcome to the deep archive. We leave the surface. The hatch seals. Below us wait sealed files, internal Monsanto emails, ghost-written scientific papers, a regulatory fog where two agencies read different substances under the same name, a glass of tap water on a farmhouse counter, half full, carrying residues that were never meant to stay. The question, what happens when a perfect herbicide becomes infrastructure you cannot remove? The archive is open. The descent begins. The smell comes first. It arrives before the sound of the spray rig, before the tractor tires flatten the hedgerows. A sweet, chemical sweet odor, almost medicinal, that settles into the back of the throat and stays there for hours. In July, in the American Midwest, this is the smell of progress, of efficiency, of a crop that will not have to compete for sunlight, water, or nutrients. The smell of a herbicide that was supposed to solve the oldest problem in agriculture, the weed. But the molecule that produced that smell did not begin as a weapon against weeds. It began as a water softener. In the 1950s, the chemists at Monsanto were playing with a class of compounds called phosphonates, molecules built around a phosphorus atom bonded directly to carbon, a structure that nature rarely uses. They were investigating them as chelators, substances that grab metal ions out of solution, binding calcium and magnesium so that hard water turns soft. The compound that would later be called glyphosate was the third iteration of that research, a molecule refined for its ability to grip minerals. It was effective. It was also forgotten, a laboratory curiosity filed away in a drawer. Then, in 1970, a Monsanto chemist named John Franz decided to test it on plants. He was looking for herbicides, and he had a hunch that the molecule's metal-binding talent might do something inside a living cell that simple poisoning could not. He was right, but even he did not understand exactly what the molecule was doing. The strange part was not that it worked. Many compounds kill plants. The strange part was that it killed everything green with a precision that seemed almost intelligent. The mechanism took years to unravel. The molecule entered through the leaves, traveled through the vascular system, and then did something no other herbicide had done. It attacked a metabolic pathway that plants share with bacteria and fungi, but that animals, including humans, do not possess. The Shikimate pathway. Biochemists had only mapped it in the 1950s, and it remained an obscure footnote in metabolic textbooks. It produces 3 essential amino acids: phenylalanine, tyrosine, and tryptophan. Without them, a plant cannot build proteins, cannot synthesize hormones, cannot defend itself. It starves from the inside. Glyphosate blocked the 5th enzyme in that pathway, a protein called EPSPS. The inhibition was irreversible. The plant didn't wilt or rot, it simply stopped growing. The leaves turned pale, then yellow, then brown, and within 2 weeks the weed was dead. Its own metabolism turned against it. And because mammals do not have the Shikimate pathway, the molecule passed through the body unaltered, excreted in urine, leaving no trace. That was the argument. That was the promise. Monsanto understood immediately what they had. A herbicide that attacked a target humans did not have, that broke down in soil within days, that could be sprayed over crops without fear of residue. It seemed like a surgical strike in an industry that had only known blunt instruments. The patent was filed in 1971. The product was branded Roundup in 1974. And within a decade, it was being sprayed on millions of acres across the United States, from the cotton fields of the Delta to the corn rows of Iowa to the suburban lawns of California. The lawn was a deliberate strategy. In 1976, Monsanto repackaged the agricultural chemical for homeowners, selling it as a precise, easy-to-use solution for the idealized American yard. The suburban homeowner, standing at the edge of his driveway with a hose end sprayer, would apply the same molecule that farmers used on thousands of acres, but he would apply it far more heavily because his equipment had no calibration, no nozzle pressure regulator, no sense of dosage. The lawn, that symbol of domestic order, became a site of chronic, low-dose exposure. The molecule that was supposed to disappear into the soil was instead accumulating at the edges of houses, next to driveways, under the windows of bedrooms where children slept. And still, no one asked the question that would haunt the molecule decades later. The question was not whether it killed weeds. It did that beautifully. The question was, what happened when you sprayed something so effective that you no longer needed to rotate crops, till soil, or pull weeds by hand? The old agriculture had built soil structure through disturbance and diversity. Plowing aerated the ground, crop rotation broke pest cycles, manual weeding kept any single species from dominating. All of that was invisible labor, measured in sweat and time. Glyphosate replaced it with chemistry. The molecule became infrastructure. The problem with infrastructure is that it is never questioned until it fails. By the late 1980s, the first cracks appeared. Weeds began to survive spraying, not through avoidance. They were hit with the same chemical, but through evolution. A few plants in every field carried a genetic variation that made their EPSPS enzyme slightly less sensitive to glyphosate. Under normal conditions, that variation gave no advantage. But when the entire field was drenched in the molecule year after year, those few resistant plants survived, reproduced, and spread. Farmers responded by spraying more. The dosage climbed. The soil absorbed the excess. And what happened to the soil was not part of the safety studies. The molecule chelated metals. That was its original purpose. And in the soil, it bound to manganese, zinc, copper, iron, making them unavailable to crops. The fungi that formed symbiotic networks with plant roots changed in composition. The bacteria that break down organic matter declined. The earthworms that aerate the soil became less active. The invisible world beneath the plow line was being quietly rearranged, and no one had a baseline to measure the change. The regulatory system trusted the molecule. The EPA evaluated glyphosate in the 1980s and found it safe, relying on studies that tested the pure active ingredient, not the formulated product that farmers and homeowners actually sprayed, with its surfactants and adjuvants that made cell walls permeable. Those inert ingredients were not tested for safety. They were not required to be. The law assumed that if the active ingredient was safe, the whole product was safe. That assumption would take decades to unravel. But in the late 1990s, a small European research team began looking at what happens when glyphosate residues accumulate, not in soil, but in the human body. They found glyphosate in urine. They found it in breast milk. They found it in umbilical cord blood. The molecule that was supposed to break down in days was persisting, not in the environment, but inside the very bodies it was designed to leave untouched. The Shikimate pathway argument had never considered accumulation. It had never considered the surfactants. It had never considered that the perfect herbicide might leave its mark not on the plant, but on the people who lived downstream from the spray. The archive of evidence was still thin in those years. A few studies, a few researchers, a growing unease. But the molecule itself was indifferent. It did not know it was becoming a legal problem, a regulatory battleground, a symbol of everything that had gone wrong with industrial agriculture. It simply continued to do what it was designed to do: bind to minerals, block an enzyme, kill a weed. Every spring, the sprayers rolled across the fields. Every summer, the lawns were treated. Every fall, the residue levels were measured, and the levels were always low, always within limits, always deemed acceptable. The smell of a freshly sprayed soybean field in July, that sweet, chemical sweet, almost medicinal odor, became the background scent of American agriculture, so familiar that it stopped registering as a smell at all. And still, the central question remained unasked, not whether glyphosate was safe. That would be the question of the courtroom. But what happens when a perfect herbicide becomes invisible infrastructure, when the soil itself depends on it, when the food system cannot function without it, and when the only way to find out if it has damaged the world is to stop spraying and watch what grows back? No one was ready to stop. No one was ready to watch. So the spraying continued, year after year, and the molecule did what molecules do. It persisted. It accumulated. It waited for someone to ask the question it had been designed to avoid. The lawn care truck pulls up at 6:47 a.m. The engine idles low, a diesel hum that vibrates through the asphalt and into the foundations of the houses on either side. A man in a white Tyvek suit steps out, uncoils a hose, and begins to walk the perimeter of a lawn dotted with dandelions. The mist is fine enough to be invisible until it catches the early light, a gauze of droplets that settles on every blade, every clover, every crack in the sidewalk. From the street, it looks like a gardener watering, but the odor that follows, sweet, chemical, medicinal, is not the smell of rain. That bottle in the back of the truck contains glyphosate, the same molecule that had been plowed into American agriculture for nearly 2 decades. But its presence here on this suburban street was not an accident. It was a deliberate act of repositioning. Monsanto had spent the 1960s and early 1970s selling Roundup to farmers as a tool for clean fields, a herbicide that killed almost everything it touched. But the real money, the company realized, was not in the acres of soybeans and corn. It was in the millions of small, private patches of green that Americans had been taught to covet, the lawn. In 1976, Monsanto launched a consumer version. The packaging was smaller, the instructions simpler, the marketing aimed at homeowners who wanted their yards to look like golf courses. The logic was elegant. If you could sell a farmer on a chemical that saved labor, you could sell a homeowner on a chemical that saved Saturdays. and the homeowner paid more per ounce. What the marketers did not say, what they may not have fully understood, was that the average suburbanite applied glyphosate far more heavily than the average farmer. Agricultural sprayers are calibrated, tested, adjusted to deliver a precise dose across a wide field. The homeowner's spray bottle, by contrast, had no calibration at all. It was a trigger and a tank and a thumb that could pulse as long as the user wanted. The result was a chronic, low-dose exposure across the most densely populated landscapes in the country. The lawn, that symbol of order and domestic pride, became a site of quiet chemistry. For 2 decades, the lawn was where the molecules stayed. But then came 1996, and everything changed. Monsanto introduced Roundup Ready soybeans, seeds genetically engineered to tolerate glyphosate. The plant could absorb the spray and survive. The farmer could now apply the herbicide directly over the crop, killing only the weeds. It was not a refinement of previous practice. It was a replacement of the entire system. Previously, farmers had rotated herbicides, tilled the soil to disrupt weed growth, and accepted that some manual weeding was inevitable. Now, they could plant, spray once, and walk away. The adoption was staggering. By 2007, more than 90% of U.S. soybeans were Roundup Ready. Corn followed at 85%. The molecule that was supposed to break down in soil within weeks was now being applied so relentlessly that it no longer had time to degrade between seasons. It saturated the ground, the water, the air, the crop itself. The seed had become a delivery system, and the field had become a pharmacy. The strange part was not that the system worked. It worked spectacularly at first, but that it was sold as environmentally progressive. No-till farming, made possible by chemical weed control, reduced soil erosion and trapped carbon in the ground. Farmers did not have to burn diesel fuel to plow. The topsoil stayed put. The carbon stayed in the earth. Glyphosate, the argument went, was the greenest chemical ever invented. But the accounting was incomplete. Every season, a farmer sprayed the same chemical over the same crop. The pressure on weeds intensified. The few plants that survived the application, the ones with a natural genetic resistance, lived to reproduce. Within a few years, the first glyphosate-resistant weeds appeared. They were not a theory, they were a fact of biology. And when a weed evolved resistance, the farmer had only one option: spray more glyphosate or switch to a harsher chemical. The dependency was the point, and the dependency was the trap. By the late 1990s, the molecule was everywhere, in the soil, in the water, in the food, in the body, and yet the official position remained unchanged. Glyphosate attacks the Shikimate pathway, a metabolic route that plants and bacteria need to make amino acids. Humans do not have that pathway. Therefore, the chemical cannot hurt us. The argument was elegant, biological, and incomplete. In a quiet laboratory in France, a small team led by a researcher named Gilles-Éric Seralini began to wonder what happened when the residues accumulated, not in soil, but inside living tissue. They were not the first to ask the question, but they were among the first to publish an answer the industry could not ignore. The door they opened would not close again. The lawn care truck finishes its route by 9 a.m. The hose is wound back onto its reel. The man in the white suit climbs into the cab, and the diesel hum fades down the street. The dandelions are still standing. But the molecule is already at work, invisible, inside the cells, inside the soil, inside the water that will run into the kitchen tap hours later. The lawn looks the same. The plate looks the same. The story, however, had just begun to turn. The water came from a tap in the Midwest, from a town surrounded by corn and soybeans. It tasted clean, clear, cold, the way municipal water is supposed to taste. By every legal standard, it was clean. But when a researcher ran a sample through a mass spectrometer, the machine found something the tongue could not: glyphosate, at a concentration of 2 parts per billion. Not enough to register on any regulatory scale. Enough to register on the question of whether anything we swallow is truly free of the fields. That was the early 2000s. The finding came from the Centers for Disease Control, which had begun testing human urine for environmental chemicals as part of a broader biomonitoring program. The first national report on human exposure to environmental chemicals, published in 2001, did not include glyphosate. Neither did the second in 2003. But by 2005, the protocols had expanded, and the results arrived with a quiet authority. Glyphosate was present in the urine of American adults. Not everyone, but enough. The levels were low, measured in parts per billion, concentrations that had previously been considered biologically meaningless. The shock was not the quantity, it was the existence. Until that moment, the story of glyphosate had been built on a single biological fact: the Shikimate pathway. Plants and some microbes need it to make amino acids. Animals do not. Therefore, the chemical had no target in the human body. The argument was elegant, almost surgical. It was also incomplete. The pathway was real. The absence in humans was real. But the assumption that no pathway meant no effect was not a scientific conclusion. It was a bet. And the bet was that the body's complexity would never find another way to take damage. By the mid-2000s, a French biologist named Gilles-Éric Seralini had begun to test that bet. His laboratory at the University of Caen was small, his funding modest, and his methods unconventional. He fed rats a diet containing Roundup, not pure glyphosate, but the commercial formulation that farmers and homeowners actually used. The rats developed hormonal disruptions, kidney damage, liver lesions, and mammary tumors at doses far below the official safety limits. The industry responded, as industry often does, with attacks on methodology, on the journal that published the study, on Seralini himself. His work was called flawed, his data cherry-picked, his conclusions alarmist. The retractions and counter-studies followed. And yet, beneath the noise, Seralini had touched something that the original safety studies had missed. The glyphosate molecule is never sprayed alone. It comes dissolved in a liquid that includes surfactants, chemical wetting agents that help the herbicide penetrate plant leaves. The most common of these is polyoxyethylenamine, or POEA. In the plant, POEA disrupts the waxy cuticle of the leaf, allowing glyphosate to reach the interior. In the human body, POEA does something similar. It makes cell membranes more permeable. The formulated product, the one sprayed across millions of acres and suburban lawns, contains not one active agent, but a synergistic cocktail. The pure active ingredient, the one approved by regulators, was tested in isolation. The commercial product was not, and the difference was not a detail. It was the central flaw in the safety assessment. A quiet war began in the scientific literature. It was not simply about whether glyphosate caused cancer, though that question would eventually dominate the headlines. It was about what counted as a relevant study. The studies that found no harm typically used pure glyphosate, administered in controlled doses to laboratory animals, measured over standard time frames. The studies that found harm used the commercial formulation, or they looked at real-world exposure patterns, the cumulative effect of spraying, eating, drinking, breathing a molecule that was now everywhere. The two bodies of evidence did not contradict each other. They examined different substances using different assumptions. The question was, which set of assumptions matched reality? The question was also whose studies would be trusted. The industry funded its own research, as companies do, and submitted it to regulatory agencies. The independent researchers funded their own work, or secured government grants, and published in peer-reviewed journals. Regulatory agencies tended to privilege the industry studies because they were conducted under good laboratory practice protocols, standardized, repeatable, and legally defensible. Academic studies used different methods, longer observation periods, and more sensitive endpoints. One of the two was designed to satisfy a legal standard. The other was designed to find what was actually happening. They were not the same kind of document. Meanwhile, the human biomonitoring data kept accumulating. By 2010, researchers had found glyphosate in breast milk. By 2012, in umbilical cord blood. By 2015, in the urine of children eating conventional diets, and significantly lower levels in children who ate organic. The molecule was not supposed to persist. It was supposed to break down in soil, to degrade in sunlight, to be excreted harmlessly if ingested. But here it was, passing from the field to the stomach to the bloodstream to the placenta, moving through the human body as though the Shikimate pathway argument had never been made. The laboratory studies began to suggest mechanisms that had nothing to do with amino acid synthesis. Glyphosate was shown to disrupt endocrine signaling, particularly in the regulation of estrogen and testosterone. It altered the gut microbiome, killing beneficial bacteria in the same way it killed weeds, by blocking the Shikimate pathway that gut microbes, like plants, depend on. The very pathway that made the molecule safe for humans also made it dangerous to the microbial ecosystem inside humans. The body is not a sealed container, it is a colony, and the colony was under chemical pressure. The paradox was almost architectural. The safety case for glyphosate rested on a precise biological target that humans did not possess. But the harm, if it existed, came from collateral damage to systems the human body does possess: hormone receptors, microbial communities, cell membrane integrity, kidney function, mitochondrial health. The original studies had been designed to look for a specific kind of toxicity: acute poisoning, organ failure, obvious tumors. They had not been designed to look for endocrine disruption at low doses over a lifetime. They had not been designed to look for changes in the gut flora of a developing fetus. The tests had been written for a different kind of question, and the answer they returned was only as good as the question. The strange part was not that the molecule could cause harm. It was that the harm, if real, was subtle enough to hide inside the normal variation of human health. Non-Hodgkin lymphoma, the disease most consistently linked to glyphosate exposure in agricultural workers, is not a rare cancer. It occurs in the general population at a background rate of about 20 cases per 100,000 people per year. A 1.4 times increase among farm workers means roughly 8 additional cases per 100,000. Not a plague, not a catastrophe visible from the outside, but 8 people per 100,000 year after year, decade after decade, who would not have gotten sick if the fields had been managed differently. The quiet accumulation was not just chemical, it was also statistical. The numbers built up slowly, study by study, lawsuit by lawsuit, until they reached a threshold where they could no longer be dismissed as coincidence. The industry called the associations weak. The critics called them evidence. Both were correct in their own frameworks. The problem was that the frameworks were not designed to talk to each other. In 2015, the International Agency for Research on Cancer classified glyphosate as probably carcinogenic to humans. The decision was based on limited evidence in humans and sufficient evidence in animals. It was not a statement about risk at the levels found in tap water or breakfast cereal. It was a statement about a pattern. When the data were taken together, the direction of the signal was clear. The molecule was not innocent. How guilty it was, and at what dose, remained a question for other agencies, other courts, other decades. In a lab in France, Seralini continued his work, though his career had been damaged and his funding had dried up. In a field in Iowa, a farmer looked at the label on a Roundup jug and tried to decide whether the small font warnings applied to him. In a kitchen in Ohio, a mother poured a glass of tap water and did not think about the watershed upstream. The molecule was in all of them. It was in the soil, the rain, the dust, the grocery store produce, the cereal box, the lawn where children played. It had become invisible infrastructure. And the question was no longer how it got there. The question was whether the human body could keep its own accounts in a ledger it had never been asked to audit. The evidence kept arriving. The studies kept publishing. The regulatory decisions kept being renewed. And somewhere between the laboratory and the courtroom, between the mass spectrometer and the jury verdict, the story of glyphosate changed from a question of safety to a question of what it means to live with a chemical you can no longer remove. The archive was still open, but the files were no longer just about the molecule. They were about the people who sprayed it, ate it, drank it, breathed it, and waited for the body to show its work. The fluorescent lights hum at a frequency just below hearing, a constant low thrum that fills the hallway of the San Francisco County Courthouse like invisible water. On the tile floor, the sound of leather shoes echoes, deliberate, measured, the gait of lawyers carrying cardboard boxes. Each box holds paper, memos, emails, study reports, internal risk assessments, decades of corporate history reduced to portable rectangles. The lawyers do not run. There is no urgency in their stride. The urgency happened years ago, in laboratories and fields, in the bodies of men who sprayed a chemical they were told was safe. Now the boxes are evidence. The hallway becomes an archive of a different kind, not of published science, but of what was said behind closed doors. In March 2015, the International Agency for Research on Cancer, a branch of the World Health Organization headquartered in Lyon, France, classified glyphosate as probably carcinogenic to humans. The designation was based on limited evidence in humans, primarily non-Hodgkin lymphoma among agricultural workers, and sufficient evidence in animals. The announcement did not land like a verdict. It landed like a spark in a room full of dust. Within hours, the chemical industry issued statements calling the classification a statistical anomaly, a methodological error, a conspiracy of anti-chemical ideologues. The industry pointed to other agencies: the United States Environmental Protection Agency, the European Food Safety Authority, the German Federal Institute for Risk Assessment, all of which had concluded that glyphosate was unlikely to cause cancer under normal conditions of use. The contradiction was real, and it was unresolved. The science was not settled. It was fractured, not because the data were missing, but because the same data were read through different lenses. The EPA weighed studies of pure glyphosate. The IARC weighed studies of the formulated products that farmers and groundskeepers actually sprayed. The two agencies were not analyzing the same substance, though both used the word glyphosate. The molecule itself became a Rorschach test. See it as pure active ingredient, and it appeared benign. See it as the mixture sold in bottles with surfactants and adjuvants that make human cells permeable, and the picture darkened. The contradiction might have remained a footnote in the regulatory archives, if not for a groundskeeper named DeWayne Johnson. In his 40s, Johnson had worked for the Benicia Unified School District in California, spraying Roundup on school grounds as part of his daily routine. He wore a short-sleeved shirt. He did not wear a respirator. The label said the product was safe when used as directed. In 2014, he was diagnosed with non-Hodgkin lymphoma, a cancer of the lymphatic system, the body's fluid network, the channels through which immune cells travel and chemical residues filter. He was 46 years old. He had no family history of the disease. He had, however, sprayed glyphosate on hundreds of acres over the course of his employment. Johnson sued Monsanto, alleging that the company knew about the cancer risk and failed to warn him. The case went to trial in San Francisco in 2018. The trial was not a referendum on glyphosate's safety in the abstract. It was a question of knowledge and concealment. The plaintiff's attorney subpoenaed internal Monsanto documents, emails, meeting notes, draft studies. What the jury saw was not scientific debate, but corporate behavior. They saw emails in which Monsanto employees discussed ghostwriting scientific papers, drafting manuscripts that would be attributed to academic researchers who had not written them. They saw communications about interfering with regulatory reviewers at the EPA, attempting to shape the agency's conclusions before they were published. They saw a campaign to discredit the IARC after the 2015 classification, not by engaging with the data, but by attacking the scientists, their methods, their motives. One internal memo proposed a major push to discredit IARC's work on glyphosate by questioning the validity, accuracy, and objectivity of the assessments. The jury was not asked to decide whether glyphosate caused DeWayne Johnson's lymphoma. That is not how product liability law works. The question was whether Monsanto knew or should have known that the product posed a risk, and whether it failed to warn users. The evidence of concealment was, in the jury's eyes, sufficient. They found Monsanto liable and awarded Johnson $289 million in damages, later reduced on appeal, but the message remained. The courtroom became a laboratory of a different kind, not a place where molecules were tested, but where the history of those molecules was tested. The jury did not settle the science. They settled the question of whether the company had acted in good faith. The verdict said no. The significance of that verdict extended far beyond one man's case. In the years that followed, the litigation multiplied. By 2024, Bayer, which had acquired Monsanto in 2018 for $63 billion, had settled over 100,000 lawsuits for more than $10 billion. The company continued to insist that glyphosate was safe, that the regulatory approval stood, that the science on their side was sound. Yet the settlements were an admission of a different kind, not of guilt, but of the cost of prolonged denial. The legal system, not the scientific one, had become the venue where the question of safety was adjudicated. Juries in California, Missouri, Pennsylvania, and elsewhere reached similar conclusions. The company had hidden what it knew. The verdicts carried no regulatory weight. The EPA still considered glyphosate safe for use, but the gap between what is legal and what is condemned widened until it became a canyon. Consider the shape of that canyon. On one side, regulatory agencies operate under procedural rules designed to evaluate products before they reach the market. They rely on studies submitted by the manufacturer, studies conducted according to standardized protocols that test the active ingredient in isolation. On the other side, juries evaluate the lived experience of people who used the product as it was sold, the mixture of glyphosate with surfactants, the cumulative exposure over years, the real-world conditions that no controlled study can fully replicate. The two systems speak different languages. One asks, is this chemical safe under ideal conditions? The other asks, did this company lie about what it knew? The answers do not align. They cannot align because the questions are different. The fluorescent light in the courthouse hallway does not flicker. It casts a flat, even glow on the tile, on the boxes of documents, on the faces of lawyers who have spent years reading emails written by men who believed the archive would never be opened. The emails are still there. The molecule is still there. It is sprayed on fields in the spring, on lawns in the summer, on school grounds where children play. The legal verdicts do not stop the spraying. The regulatory approvals do not stop the lawsuits. The contradiction persists, unresolved, because the question it raises has no comfortable answer. If a substance can be simultaneously legal and condemned, if it can be approved by the state and found dangerous by the citizenry, then safety is not a property of the molecule at all. Safety is a negotiation. It depends on who is asking, how soon the answer is needed, and whose body will bear the cost of being wrong. The empty container lies in the ditch, half buried in last season's dead grass. The label is still legible. Keep out of reach of children, in English on one side, Spanish on the other, beneath a diamond-shaped hazard symbol that looks like an abstract warning nobody has time to study. The plastic is bleached by months of sun, the green of the brand name faded to a pale ghost. The container once held the solution itself, the formulated product, not the pure molecule. But nobody asked that distinction when the sprayer emptied it. The label told the worker what to wear. It did not tell the regulator what to measure. That difference between the chemical on the label and the chemical in the field is the crack through which the entire regulatory debate has slipped. For decades, the Environmental Protection Agency evaluated glyphosate the way a pharmacist tests a raw ingredient, pure, isolated, stripped of its companions. The agency required studies of the active substance, the white crystalline powder that John Franz first identified in his Monsanto laboratory in 1970. The agency did not require equal testing of the formulation that farmers actually filled into their tanks. The liquid that contained glyphosate along with surfactants, wetting agents, preservatives, and a proprietary blend of compounds designed to help the molecule penetrate plant cell walls. That mixture, the one that smelled of medicine and clung to the back of the throat, was tested only by the manufacturer, and those tests were not submitted to the EPA for the same rigorous review. The International Agency for Research on Cancer, when it classified glyphosate as probably carcinogenic to humans in 2015, did not make that distinction. It looked at the real-world exposure, the formulated product, the spray drift. the residues on crops, the occupational contact that farm workers and groundskeepers experienced every season. IARC reviewed the epidemiological evidence built not from lab beakers, but from blood samples and hospital records, and years of spray logs kept in farmhouse drawers. Two agencies, both using the same word, glyphosate, were analyzing different substances. One evaluated a molecule. The other evaluated an exposure. The contradiction that erupted in headlines was not a failure of science. It was a failure of definition. The strange part was not that the two bodies disagreed. It was that the disagreement had been visible for years before anyone forced it into public view. The EPA had long maintained a policy that when it reviewed pesticides, it evaluated the active ingredient alone. The formulated product was considered a trade secret, its full composition protected as confidential business information. So the agency that was supposed to protect the public from chemical harm was reading only half the recipe. The scientists who studied the whole product, who mixed the tank and analyzed the spray, were published in journals, not regulatory dockets. Their findings existed, but they belonged to a different archive. The European Union took a different path. Not because European regulators were more virtuous. They had their own pressures, their own industry ties, their own history of agricultural chemical use, but because the institutional architecture forced a different question. The European Food Safety Authority asked not, is this chemical safe in isolation? But is this chemical safe under the conditions it will actually be used? The answer was a managed compromise. In 2023, when glyphosate's license renewal came before the European Commission, the approval carried restrictions that would have been unimaginable in American regulatory practice. Aerial spraying, routine over vast American fields, was banned outright. Buffer zones of 5 to 10 meters were required along waterways and residential edges. The practice of desiccation, spraying glyphosate on grain crops just before harvest to dry them for earlier collection, was prohibited entirely. The chemical was not declared unsafe, it was declared controllable. The message was subtle but decisive. The European judgment said, "We do not trust this substance to be used anywhere, anytime, in any quantity. We trust only a system that monitors, limits, and mitigates." The American message, embedded in a century of land management culture, said the opposite. The molecule is safe until proven otherwise, and the burden of proof belongs to the injured, not the manufacturer. That fundamental asymmetry is encoded in the regulations themselves, in the fine print that nobody reads, just like the label on the empty container in the ditch. The American model is built on permission. Once a chemical is registered, it is presumed acceptable, except where explicitly restricted. The European model is built on precaution. The more it is used, the more it must be watched. The same molecule, the same metabolic pathway it blocks, the same residues that accumulate in soil and water and human tissue. But safety became a property, not of chemistry, but of jurisdiction. What was safe in Iowa required gloves and a respirator in Normandy. A farmer in Nebraska could spray from the air without notice. A farmer in Bavaria needed signs posted and a buffer of bare earth before the chemical touched the ground. The consequences are not abstract. They show up in the bodies of those who do the work. In the United States, agricultural workers have higher measured concentrations of glyphosate in their urine than any other population group, and higher than workers in European countries with stricter exposure limits. The American farm worker is not protected by the label. The label is a legal document, not a shield. The European farm worker operates under mandatory training, prescribed equipment, and post-application waiting periods that limit re-entry into treated fields. The difference is not the chemistry. The difference is what a society is willing to accept as the price of a weed-free field. And yet both sides face the same uncomfortable truth. Glyphosate is now embedded in the global food system at a depth that makes withdrawal almost unthinkable. Remove it, and the no-till agriculture that has saved billions of tons of topsoil from erosion collapses. Remove it, and the Roundup Ready seed system, engineered for a world of glyphosate-dependent farming, loses its economic logic. Remove it, and the cost of food rises. The complexity of weed management returns. The machinery of industrial agriculture must be redesigned. The chemical is not just a product. It is infrastructure, invisible, underground, but structural to the way the world eats. So the regulatory fog persists, not because the science is unsettled, though it is, but because the decision is too expensive. The European Union chose monitor and limit. The United States chose trust and use. Both choices are political, not scientific. Both choices are made by people who will not be the ones spray drifting across a soybean field at dawn. The empty container in the ditch will eventually be collected by a farm worker, by a county cleanup crew, by rain that washes it downstream, by a child who mistakes it for a toy. The label will fade further. The hazard diamond will become unreadable. But the question the container asks does not fade. Who decides what safe means, and why does the answer depend so much on where you stand? The regulatory battle will continue. More studies will be published, more lawsuits will be filed, more renewals will be negotiated. But underneath the hearings and the position papers and the conflicting meta-analyses, the problem is not that glyphosate is dangerous. It is that we built a world that requires it. The molecule did not demand this dependency. We did. And now, the only question left is whether we can afford to stop and whether we can afford not to. The archive does not close on that question. It remains open, waiting for an answer that might not arrive until the last container is buried in the last ditch, and the last label is too faded to read. The silence of a farmhouse kitchen at midnight is a particular kind of quiet. The refrigerator hums. The clock on the microwave ticks. A glass of tap water sits on the counter, half full, the way it does in a house where someone will be back in a few hours to finish it. Outside, a distant irrigation pump cycles on in the dark, a low mechanical heartbeat across the field. The water in that glass came from the ground beneath that field. It passed through soil that has been sprayed year after year, season after season, with a molecule that was supposed to break down and disappear. But the molecule did not disappear. It learned to linger. The long-term ecological consequences of glyphosate are only now becoming visible, not because the evidence was hidden, but because nobody thought to look at the invisible. The early safety studies had asked the obvious questions. Does it kill rats? Does it cause cancer in lab animals? Does it persist in soil beyond a few weeks? The answers were reassuring enough. The rats survived. The cancer rates did not spike. The half-life in soil was measured in days, not years. The molecules seemed to vanish. But vanishing is not the same as absence. What the early studies missed was the effect on the world that exists in the spaces between roots, in the films of moisture around soil particles, in the microscopic communities that underpin the entire system. Glyphosate does not just kill weeds. It alters the soil microbiome. It reduces populations of beneficial bacteria that plants depend on for nutrient exchange. It chelates essential micronutrients like manganese, binding to them and making them unavailable to crops. The soil becomes chemically poorer, biologically simpler. After years of treatment, the fungal communities shift. The earthworm populations decline. The invisible infrastructure of the field is rewritten, one molecular signature at a time. The strange part is not that glyphosate does this. Any broad-spectrum biocide will disturb an ecosystem. The strange part is that the safety tests never required measuring these effects. The EPA evaluated the active ingredient in isolation. The commercial product, the one actually sprayed, contains surfactants that amplify the damage. And the long-term studies, the ones that would track a single field across a decade, were never mandatory. The result is a slow-moving ecological debt, one that doesn't appear on any ledger until the yield begins to drop, the soil begins to compact, the crops begin to show micronutrient deficiencies that cannot be explained by any other cause. By then, the invoice is already overdue. The human health question remains unsettled in a way that feels deliberate, almost architectural. The epidemiological studies have been accumulating for years. Meta-analyses of the best available data consistently find a modest but statistically significant association between glyphosate exposure and non-Hodgkin lymphoma. The relative risk is about 1 for a 40% increase over the unexposed population. In clinical terms, that is not a powerful carcinogen. A smoker's risk of lung cancer is many times higher, but there is a trap built into that comparison. The effect of a weak carcinogen depends entirely on the scale of exposure. When the entire agricultural workforce is drenched in a chemical, when the residues drift into suburban lawns, when the molecule appears in the urine of children who have never touched a sprayer, a 40% increase in risk translates into thousands of excess cases. The numbers are not abstract. They have names. They have court dockets. The mechanism remains disputed, but the pattern is suggestive. Glyphosate is not supposed to affect human cells because we lack the Shikimate pathway. But that argument assumed the only way the molecule could cause harm was through that specific biochemical lock. The body has many locks. Glyphosate formulations disrupt endocrine signaling. They alter the gut microbiome. They increase the permeability of cell membranes. The surfactants see to that. The molecule is not a key that fits a single door. It is a key that fits locks we did not know existed, locks that open into rooms we had not yet mapped. The regulatory agencies have handled this uncertainty differently. The EPA considered the pure active ingredient and found it safe. The International Agency for Research on Cancer considered the formulated product and found it probably carcinogenic. Two agencies, looking at overlapping data, reached opposite conclusions. The divergence was not a failure of science. It was a disagreement about what counts as evidence, about whose studies are trusted, about whether the burden of proof falls on those who suspect harm or those who assert safety. The European Union, after years of debate, renewed glyphosate's license in 2023, but wrapped it in restrictions: no aerial spraying, buffer zones near water and homes, a ban on using the chemical to dry crops before harvest. The message was not that glyphosate was safe. It was that it could be managed, if you accept that management means accepting a certain level of exposure. The American approach had been simpler. Trust the manufacturer, spray freely, let the market decide. The European approach was a compromise, a treaty signed between agriculture and caution. Neither side declared victory. Neither side could. We have built a food system that depends on a chemical whose long-term effects we do not fully understand. Glyphosate is embedded in the global agricultural infrastructure. Roundup Ready soybeans, corn, cotton, canola. Over 90% of U.S. soybean acres are planted with seed engineered to survive the spray. The chemical is applied not once, but multiple times per season, sometimes as a pre-plant burn-down, sometimes as a post-emergence treatment, sometimes as a desiccant to dry the crop before harvest. Residues accumulate in grain, in soil, in water, in the bodies of farm workers and consumers. Withdrawing glyphosate tomorrow would cause massive agricultural disruption. The no-till farming practices that depend on it would collapse. Weed resistance, already a growing problem, would accelerate. Yields would drop. Food prices would rise. The system has no graceful exit. And yet the same system is generating a steady stream of legal, scientific, and regulatory challenges. By 2024, Bayer had settled over 100,000 lawsuits for more than $10 billion. The company continues to insist the chemical is safe, even as it pays billions to people who claim the chemical made them sick. The contradiction is not lost on the juries. They are not asked to decide the science. They are asked to decide whether the company hid what it knew. And the internal documents, the emails, the ghostwritten papers, the regulatory interference, those are facts, not interpretations. The jury system, imperfect and provincial, has become the venue where the question of glyphosate's safety is effectively adjudicated, because the regulatory system could not resolve it. The Deep Archive does not close this case. It files it under unresolved. Not because the evidence is lacking. There is plenty of evidence, too much, pointing in contradictory directions. The archive remains open because the story is still being written. Every spring, the sprayers roll across the fields. The tractor cabs are air-conditioned now, sealed against drift. The nozzles are calibrated. The operator wears gloves and a mask, if the label is followed. The mist settles on the green leaves, and within days the weeds turn yellow, then brown, then brittle. The crop survives. The soil absorbs what it can. Every summer, the lawns are treated. The landscapers arrive before dawn, the trucks idling on quiet suburban streets. They walk across the grass, hoses trailing behind them, spraying the precise pattern that promises a weed-free July. The children will play on that grass in the afternoon. The dog will roll in it. The residue will be tracked into the house on shoes, on paws, on bare feet. Every fall, the residue levels are measured. The water samples are collected at the mouths of watersheds. The urine samples are collected in public health studies. The soil samples are shipped to labs that will test for parts per billion. The numbers are published in journals that few people read. The regulatory agencies review them. The industry scientists review them. The independent researchers review them. And the debate continues, because the numbers never settle into a single story. Every year, another lawsuit is filed, another study is published, another regulatory decision is made. Another family receives a diagnosis and begins to wonder whether the years of spraying, or living next to the field that was sprayed, or drinking the water that ran through the sprayed soil, had anything to do with the cells that are now dividing where they should not. The question glyphosate leaves us with is not whether it is safe. The question is whether we are comfortable living in a world where we will never fully know. The archive remains open. The glass of water sits on the counter. The irrigation pump cycles on in the dark. And somewhere, in a farmhouse kitchen very much like this one, the spray schedule for tomorrow has already been written on a calendar pinned to the wall. The paradox that surfaced from this descent was not that glyphosate kills weeds. It was that the molecule designed to vanish became infrastructure. The empty container in the ditch with its fading label. The glass of tap water in the farmhouse kitchen, drawn from soil sprayed season after season. The cardboard boxes of internal emails carried through a courthouse hallway, evidence of what the company knew and chose not to share. What the descent revealed was that safety is not a property of a molecule. It is a negotiation between the bodies that absorb it and the institutions that approve it. The cost of progress became visible only when the residue could no longer be filtered out of the story. Now the pressure eases from the hull. The darkness thins above, black water turning blue. The hatch opens, and cold sea air enters the lungs. The archive file closes, but the recovered evidence remains with us. Some substances are safest when they remain evidence, not experience. If this kind of quiet descent into science and history is what you came for, you can subscribe to the Deep Archive. The molecule is still in the field. The water is still on the counter.